Hello. Voice is not coming.
Am I audible?
Hello.
Yes. Yes, sir. Yes,
Okay.
sir. Now is audible.
Now I am audible, na?
Yes, sir.
Okay.
Yes, sir.
So can you
Yes, sir.
hello? Can you hear me?
Yes,
Yes,
sir.
sir.
Okay. Thank you. Okay. So let's start our today's session. Assignment 2. Today we will focus on the problems which are mentioned in the assignment 2. So let's start with the first problem. So what it is saying,
Voice is not coming, sir.
no, I am not saying anything.
Okay.
So it is first question is our true false based question. So it is asking that intracellular receptors are single proteins. They contain a binding region for DNA near the middle of the protein, and they are activated by hydrophobic molecule which are extracellular messenger and cross the cell membrane to reach their target. So it is true or false?
True, sir.
Yes, true. So can you explain it briefly that what is happening here? Okay. So yes, this statement is true. So intracellular receptors are the single protein. They contain a ligand binding site near the C terminal end, and they contain a binding region for DNA near the middle of the protein, and they are activated by the hydrophobic molecule. The molecule should be hydrophobic, which are extracellular messenger, and crosses the cell membrane to reach their target. Okay, so this statement is true, right? So this is true. Okay, so let's move to the next question. Okay, so in next question we are having that the enzyme chymotrypsin contains serine, histidine, aspartic acid catalytic triad. Right. So this is true or false? At first, you tell me that what is enzyme? What is the function of enzyme? Enzyme. At first, you tell me. Anybody?
Biological catalyst.
Biological catalyst. Exactly. And what is the chymotrypsin? What does chymotrypsin? It,
So chymotrypsin is a amino acid.
no, chymotrypsin is not amino acid. Chymotrypsin is an enzyme which
And
catalyzes the breakdown of the peptide or protein. So from name itself, we can get to know that it works on the protein. Like there are various types of enzymes. Some enzymes works on the some on the peptide protein, some on the carbohydrate, some on the fat. So from name, chymotrypsin, which works on the protein, right? So it is saying that the enzyme chymotrypsin contains serine, histidine, aspartic acid, a catalytic triad. So is this true or false? Hello.
True.
Yes, this is true. So the chymotrypsin contains a collection of the three amino acid called catalytic triad, as it calls the three amino acid. So it is called the catalytic triad. This triad consists of the serine 195, histidine 57, and aspartate 102. Okay. So these amino acids work together to carry out the catalytic function for breaking peptide bonds. Okay. Is this clear? So this statement
Yes,
is
sir.
true. So basically chymotrypsin, what it is does, there are various types of enzyme specific for the protein or the peptide, like chymotrypsin or proteinase K. They cut down the large protein or the peptide into a small portions. So this is what chymotrypsin do. And these three amino acid triad, which is very much specific, serine, histidine, and aspartic acid, they are very specific for this work, and sorry, work together to carry out the catalytic function. Okay. Okay, let's move to the next question. Okay, here it is saying that the CAMP, and the C stands for what? What does C signify? C means, this C, cyclic. Okay. So cyclic AMP is a neurotransmitter. So can you briefly say that what is neurotransmitter? In very uh, superficial language, what does it mean, neurotransmitter?
It's saying
which are
it
released
can
from
send the signals.
neuron
One by
nerve
one,
endings.
one by one, please, one by one.
The
Anybody?
neurotransmitters which release from the nerve endings.
Yes, nerve ending, and what it is function?
Sending the
It
signals.
exactly, sending or transmitting the signals through the nerves, right? Okay, so in our question statement, it is saying that the cyclic AMP is a neurotransmitter. Is this true or false?
False.
False. So what does CAMP do? What is its role? Okay, I have noted here some role of the cyclic AMP. This statement is false, definitely. This is not a neurotransmitter. So the chief role of cyclic AMP in several tissues seems to be facilitate or promote the mobilization of glucose and fatty acid reserves. Okay, in the liver, glucagon and the catecholamines cause an increase in the intracellular level of the CAMP by stimulating adenyl cyclase. Okay, just additional information, but CAMP is not a neurotransmitter. Okay. Okay, so let's move to the next question. Okay, so here a structure is given, alkyl ammonium ion. The nitrogen has its positive charge, and it is coordinated to the three hydrogen, and the other portion is alkyl group is there. So it is saying that labeled in D in alkyl ammonium ion is likely to be the strongest hydrogen bond donor. Okay, so we have to mark out whether the statement is true or false. This is true or false?
True.
True, yes. And why? What is the cause behind it's true?
Since nitrogen is already having three hydrogens, it is already positively charged.
Positively charged, yeah.
So
So it
the
is a better
hydrogen
strongest hydrogen bond donor, right? It is already having a positive charge. Okay, so here as we can see that nitrogen is tetra coordinated, already having a positive charge, and here hydrogen bonds are formed between a hydrogen atom bound to a small highly electronegative atom and another small highly electronegative atom with an unshared electron pair. The elements that usually participate in hydrogen bonds are nitrogen, oxygen, fluorine, that we already know that the electronegative atoms are required for the hydrogen bond, and the best hydrogen bond donor will be the one where the hydrogen is most electron deficient. So here hydrogen is electron deficient or electron rich? This hydrogen bond, this one is electron deficient or electron rich?
Electron rich, sir.
Here already it is having a positive charge in, so it is how can it be electron rich?
Sorry, sir. Sorry, sir.
So it is electron deficient system, right? And the best hydrogen bond donor will be the one where the hydrogen is the most electron deficient. In the ammonium ion, that is in our case, the nitrogen has a positive charge, and this in turn will make the attached hydrogen electron deficient. So this is why, and this N is strongest hydrogen bond donor. Okay, so this statement is true, right? Anybody, any doubt, any question? Is this clear?
Yes, sir. Clear.
Okay, so let's move to our next question. Okay, so here it is, a statement is given, that is the Michaelis-Menten equation relates the rate of an enzyme catalyzed reaction to the substrate concentration. So what is Michaelis-Menten equation? Means what it is related to? It's related to the rate expression of what? Anybody? Means what it is about, the Michaelis-Menten equation, Vmax, Km. It is related to one, means what topic? At least you should say this. It is related to the rate expression of enzyme catalyzed reaction. Right? Have you ever seen this Michaelis-Menten equation?
No.
You have not heard about Michaelis-Menten equation? Okay, so this is a equation which is related to the, basically it is a rate expression of enzyme catalyzed reaction, and which is a equation, and it states that the Michaelis-Menten equation is the rate equation for a one substrate enzyme catalyzed reaction, This equation relates the initial reaction rate V0, the maximum reaction rate Vmax, and the initial substrate concentration S through a Michaelis constant Km, a measure of the substrate binding affinity. So this statement is true or false? In our statement we are getting the Michaelis-Menten equation relates to the rate of an enzyme catalyzed reaction to the substrate concentration. So is this true or false? Here substrate concentration is here. We are getting substrate concentration, and we are also getting the rate, reaction rate, right? So is this true or false, this statement? Hello. Yes,
True, sir.
this is true. So from question, just see what it is asking for. It is asking that the Michaelis-Menten equation, rate of an enzyme, here rate is there, the rate of an enzyme catalyzed reaction, and how it is related to the substrate concentration. And in the explanation, we are also getting that the reaction rate, that is V, and the substrate concentration S is also there. So basically, we can get a correlation in this expression. So this statement is true. Okay.
Okay, sir.
Okay. So let's move to the next question.
Yes, sir.
Okay, here reaction mechanism is given. So we have to say that what kind of reaction is this? Can anybody know this? Can anybody tell me what kind of reaction is this? Is this anchimeric assistant or oxidation or reduction or hydrolysis? What is happening here in this reaction?
Antimeric questions, sir.
NGP,
Exactly.
neighboring group participation.
Yes, neighboring group participation, NGB or anchimeric assistance, right? So let's see
Yes.
how it is happening, taking place. So here S, and here chloro is there. So sulfur is having lone pair. So what will sulfur do now? Sulfur will attack
Carbon atom and leave
Orange,
chlorine.
sir.
and leave chlorine, right?
Chlorine.
Yes. So here a 3-member ring will be formed. Right. So now alkoxide is there. So alkoxide will attack here or here. Which place?
First.
Anyway, both are identical. It can attack here or here. The same thing will happen. So if it attacks here, this carbon, this will be opened up. And we are getting, okay.
No.
So why are we calling it at NGP, neighboring group? Why the name is justified, that neighboring group participation?
For the departure of leaving group, neighboring group.
Yes, what we see in various substitution reaction, that various kind of alkyl halide, or many cases that backside attack is there from the other nucleophile, from the solution is attacking that atom. And here we are seeing that the S, the sulfur, that is here, it is a new acting as the nucleophile, and chlorine is the leaving group. They are in the same molecule, right? They are in the close proximity. So the rate is very much higher. As the sulfur is very much close proximity to this carbon, so the rate of attack of the sulfur to the carbon atom is very much faster and very in very good amount. Okay, so this is why this name comes from the neighboring group, that sulfur is the neighbor of the carbon that is in very close proximity, and it is attacking to the carbon atom to facilitate the substitution process.
We get
Okay.
retention of configuration.
Yes, we get retention. So fast, this is the first attack, and here also the second attack is going on. So we are getting retention of configuration, right?
Yes, sir.
Okay. So anybody, any doubt in this question?
No, sir.
Okay. So can we move to the next question? Okay. So let's move to the next question. Okay. So here it is saying that which of the following statements are true about the zinc finger motif. So are you aware of the term zinc finger motif?
No, sir.
Okay, so this is a motif, and it is specifically seen in the various kind of peptides or proteins. So they have various characteristics. So some of are given here, that these are DNA binding proteins. They contain cysteine residue. Cysteine residues play a very crucial role in stabilizing the zinc ions, and all of the above statements are correct. So zinc finger motifs is a special kind of motif found in the peptide or proteins. So there are some specific characteristics of the zinc finger motif. The zinc finger domain is one of the most frequently utilized DNA binding motif found in eukaryotic transcriptional factor. So this is our first point, that these are DNA binding proteins. So this is true, right? DNA binding motif. So this is true. And secondly, in second point, what we are getting, a zinc finger is a small functional independently folded domain that coordinate one or the more zinc ions to stabilize the structure to the cysteine or the histidine residue. So zinc finger motif, there are various kind of amino acid residues are there. So cysteine and histidine are the very much important for the binding. So these two later two statements are also correct, right? They contain cysteine residue, they definitely contain cysteine residue. And the third one is saying that the cysteine residue play a very crucial role in stabilizing the zinc ions. So yes, this is also true. Cysteine residues play a very good role in stabilizing the ions. So our correct option is which one?
All of the above.
All of the above are correct. So all these statements are true about the zinc finger motifs, that they are DNA binding proteins, they contain cysteine residues, and cysteine residues plays a very crucial role in stabilizing the zinc ions. Okay.
Sir, would you please explain something more about zinc finger motifs?
Okay, so zinc finger motif. This zinc finger motif is a very much. It is a very characteristic, and it is mainly DNA binding motif. This motif is specifically characterized known for the binding the DNA near DNA, and these two amino acids which is present there, and from the name itself we are getting that zinc ion is also there. That is why it is called zinc finger motif. And these amino acids which are in the question was there, that the cysteine and the histidine, they are very much crucial for this binding of the DNA, and this is specifically found in the eukaryotic cell, and this is a very crucial for the DNA binding. Okay.
Okay, thank
Okay.
you, sir.
So let's move to the next question, that have you solved this numerical? It is saying that substrate A undergoes intramolecular reaction to provide C. Okay, so from A we are getting C. and it is through the intramolecular. Okay, product C. On the other hand, substrate B and C undergoes intermolecular reaction to provide the same product C. Okay, so B plus C and it is intermolecular. So what is the order of this reaction? The first reaction, this one. Anybody?
First order reaction.
First order reaction, yes. And from question it is given that the rate constant is 220 second inverse, right? And what is the order of this reaction, the second one?
Second order.
Second order, and it is rate is also given that 1 mode inverse second inverse. So how we will solve the question, that what is the effective molarity, how it is calculated, the effective molarity?
First order divided by second order.
Yes, the effective molarity is defined as the ratio between the first order rate constant of an intramolecular reaction and the second order rate constant of the corresponding intermolecular reaction. So here we are from the first order 220 second inverse divided by 1 mole inverse second inverse. So what we are getting, 220. So which is the correct answer?
Option number 2.
Option number 2. Is this clear to everybody?
Yes, sir.
Yeah, okay. So option number 2, that is 220 molar. This is the correct option for this our numerical question. Okay, so let's move to our next question. Okay, here one assertion and was reason is given that what in the statement it is saying that lactate dehydrogenase has a 100-fold selectivity for the lactate as a substrate over malate. However, if a mutation occurs that alters an active site glutamine residues to an arginine residues, the enzyme shows 10,000-fold selectivity for malate over lactate. And the reason what it is saying, that in the mutated version of the enzyme, this amino acid has been replaced by the arginine residue, which contains a protonated glutidinium functional group. This can form an additional ionic interaction with malate that is not possible with the lactate. So, what is our answer? So as we know that the lactate dehydrogenase is very much specific for the lactate. So what is the structure of the lactate? So this is the structure of what? First one? Lactate. And this is the structure of malate. So these are the two ions, means they are the conjugate base of the lactic acid and the malic acid respectively. Okay, so what it is saying, that assertion and the reason both are correct statement, and the reason is the correct explanation for the assertion. Then assertion and the reason both are correct statement, and the reason is not correct explanation of the assertion. The assertion is a correct statement, but the reason is the wrong statement. The assertion is the wrong statement, but the reason is the correct statement. So which one is the true?
First.
Yes, the first one is the correct answer. That it is assertion is the right, that whenever lactate has the very much specificity, lactate dehydrogenase towards the lactate, but whenever there is a mutation occurs at the active site, and glutamine residue to the arginine residue, the enzyme shows, then it shows to a specificity towards the malate. Okay, and the cause is also correct. The reason is also correct here. It is given that in the mutated version of the enzyme, the amino acid has been replaced by the arginine residue, which contains a protonated glutamidinium functional group. This can an additional ionic interaction with the malate that is not possible with the lactate. As you can see that here are the two negative charges there. In comparison to the lactate, here only one negative charge is there. As you can see, that only one negative charge, but here in case of the malate there are two negative charge, right? So when this mutation occurs, this can form an additional ionic interaction. Due to the additional ion, this forms an additional ionic interaction with that mutated group, which is not possible for the lactate. So that is why this, due to the mutation, this active site shows a better, more specificity towards the malate group. Okay, so the assertion and the reason both are correct, so the option 1 is the right answer. Okay, so let's come to this our last question, today's one. Here threonine is given, then with the action of the enzyme 1 we are getting a product A, then enzyme 2 we are getting B, enzyme 3 we are getting C, enzyme 4 D, and then action of enzyme 5 we are getting leucine, right? So threonine and leucine, what are they? What is their classification? What is the main compound? What we say, threonine, leucine, what it is? It is essential
Amino acid.
amino acids. These are the example of essential amino acids. So what is our correct answer? It is feedback inhibition or approximation or covalent catalysis or all of the above.
Feedback inhibition, sir.
Feedback inhibition. So how we can
Product is inhibiting the enzyme function.
product at the last stage, the product that is leucine, that is inhibiting the fast enzyme function. It is inhibiting the enzymatic function of the enzyme 1. So what we can see, this is the product. At the same time, it is inhibiting the function of enzyme 1. So this is the example of feedback inhibition, right? This is not approximation and not covalent catalysis. Okay, so whenever which cases the product is the inhibiting the action of the enzyme 1, then we can say this is the phenomena of the feedback inhibition. Okay, any doubt, anybody?
No, sir. Go ahead.
Okay. Okay, so we had only these 10 questions for this test session. So let's end for today's session. If anybody has any doubt, you can ask me. Otherwise, we will end up today's session and we will discuss on next week with our assignment 3 problems.
Sir, I have question regarding the examination part. What is the format of question paper?
Okay, for that, I think I will refer you to the main site of NPTEL. It is already given there, or you can write a mail to the coordinating committee that how does question or the exam will happen. So they can brief you in better way regarding the question and
The
of the
question
exam
paper will
pattern.
be objective type or it will subjective or mix at all?
Uh,
Descriptive.
yes, I think it is already given in the site. So you can check once, or you can communicate to the officials regarding the examination part. Okay.
Sir, I have one more question.
Yes.
For this course, faculty is only one or many numbers of faculty is teaching to us.
For one course, I think only one faculty is assigned. TAs may be more than one, but faculty is only one, I think.
Uh, Faculty is one, but what about
Yeah.
other says?
Sorry, other
Faculty is only one. One faculty is there, right?
one faculty. Yeah. One faculty is there.
So, acha. So you are from, sir?
I am from IIT Jodhpur. I am doing my PhD here.
Okay.
Sir, I have checked on the site, but there is nowhere is mention about the written examination
Okay,
format question
then
paper.
I will suggest you that if you can write a mail towards the help desk, or if any mail ID is given there regarding your queries, so it will be better and convenient for you to know about the pattern of the examination. Okay, they can tell you more conveniently or more
Okay.
appropriate way.
But you know earlier what it was, like in previous examination, what it was.
It was mixed, I think, means objective, subjective, both was there.
Okay.
Sir,
But
I have wrote. Sir, I
yeah,
have wrote a mail to the NTPL. They said that it will be MCQ type
okay.
and yes, MCQ type online examination will be there.
Okay, then it is fine. Then it is
Okay.
absolutely fine.
Okay. Ram Dogra ji, it is multiple type question.
Yeah, exactly.
Okay. You are from which city, Ram Dogra ji?
From Nagpur.
Okay. I am from Kanpur.
Okay, nice to meet you, sir.
Okay. Then
Your
as
name, sir, please.
Brijesh Tomar. My name is Brijesh Tomar.
Okay.
Okay. I am also